So one of the links in the next lecture doesn't work, so I found a couple of other links that we'll talk about. But other than that, the slides are as they were in the remainder of that YouTube lecture that you watched for Thursday of last week, and we'll finish it out today. So what we need to talk about, what you were talking about, is at the very end of last week's lecture, you're talking about the Schwann cell, and you're talking about how it helps to facilitate action potential, speed things up. Okay? And what were the two biggest influences of the conduction speed of an action potential? Diameter and whether or not it's myelinated. Okay? I gave you a clinical example on Thursday's lecture, and that clinical example was what? Multiple sclerosis, MS. Okay, so that slide is this slide right here. So for those of you that didn't watch the lecture, this is a little bit of a bonus because I'm going to back up a couple of slides so that we can kind of more seamlessly move into today's lecture from the online lecture. So this was a study that was published in Nature Reviews Neuroscience. Okay, so this is primary literature. This isn't, just so you guys understand the difference between primary literature and your textbook? No? So the textbook is actually what we would consider almost tertiary literature. Primary literature is journal publications that are published in the peer review system. And so what that means is you do some research, you get some findings, you send it to a journal for publication. The editor gets it, smells it, looks at it, decides who he wants to send it to in this field that can have an opinion about your research and your data. And that's called your peers. And so usually it's 2 or 3, usually it's 3, minimum of 2. Peers in the field that are experts in this space review your submission and determine if it's actually should be published or not. They make a recommendation to the editor of that journal, and then the editor gets back to you. That whole process can take, after you've submitted, anywhere from 3 months to like a year. Okay? Once it's published in the primary literature, it's called a peer-reviewed publication. That's what this is. It's published in 2006. But anytime you see a peer-reviewed publication, if it was published in 2006, it was probably done in like 2004 or 2005 when the experiments took place. So the primary literature is actually probably a couple of years old after it's published. Then what happens is people read a bunch of primary literature and they write review papers on the primary literature. That would be considered a secondary literature source. And then academics come along and they read the review papers and they make a textbook. So how new is your textbook? It's pretty old, isn't it? So that's why I was like, how come they always like show this crap out of the textbook, man? I don't really care. Well, we're just trying to keep you more current. That's all. We're trying to give you stuff that's outside the textbook, that's in the primary literature, that's probably newer than the information in your textbook. And that's why you don't see a lot of stuff in your textbook that professors are putting on the screen. And, you know, there's one of 2 variations. That's one variation. The other variation is it's all the professor's research, because that's all they care about. Okay? But this is not my research. This is somebody else's. And this is showing some images. So here is a neuron. And you can see this neuron is getting less healthy over time. And this is a patient who suffers from an autoimmune disorder known as multiple sclerosis, MS. And in MS, the immune system specifically targets destruction of the myelin sheath. And it kills it. It actually also kills the cells that manufacture that myelin. And then what happens is the speed of action potential decreases. And as the disease progresses, the patient has an increasing compromised action potential speed. So their motions are slower, they're not nearly as coordinated, they have more trouble walking, okay? Could lead to things like blindness, some examples. So the reason I'm backing up is because this is going to be a test question at some level on the exam. And I want to make sure that You get it virtually and you get it in person, okay? So if you're kind of wondering, oh yeah, we talked about that already. I said it almost the same way in the spring. Well, it's an important topic, okay? So the immune system attacks the myelin and kills it, destroys it, that myelin sheath. And so what we're looking at on this slide, I got to come over this way because it's colored and it's hard to see from a parallax. And I know I've learned that we've got a couple students that can't discriminate some of the colors, so I'm gonna highlight them for you. So here you can appreciate that here's a piece of myelin and here's a piece of myelin, and in between myelin are these nodes, the nodes of Gandier. And where those nodes are where your channels are located. And so at this location is where you're going to move ions. And you get a depolarization event. Remember that term? And you get a depolarization event here, and then that slightly negative signal and its depolarized result, meaning it's slightly positive, it travels underneath the myelin inside the cell, and then it exchanges ions right here again. And then it travels underneath the myelin and exchanges ions right here. So in this way, we have what type of conduction? Continuous or saltatory was the other term. Okay? So saltatory conduction is where the action potential appears to skip from one node to another, but it skips within the cell. Now what happens as the myelin degenerates is the body tries to compensate by inserting. You see where the myelin is lost in MS? Here, you've inserted more sodium-gated channels to try to compensate for that. So your body actually tries using compensatory mechanisms to make up for the lost myelin. And this is kind of this latency period of MS as the patient kind of levels off with its symptoms, or their symptoms. And then eventually, the rest of the myelin is attacked, and the body's ability to upregulate the insertion of additional sodium voltage-gated channels is maxed out. Okay? What's shown over here is immunohistochemistry. And so you can see in this top picture, can you appreciate on the left and the right here, is sort of this ghost white-looking color, and that's the myelin. And right smack dab in the middle, we have a red sphere here, where my arrow or my pointer is, and then two green-colored structures on either side. This structure is considered the voltage-gated sodium channel. And you can appreciate that it's very punctate, I mean it's right in one spot. It's not smeared across to the left and to the right, but as this disease progresses, and these two panels are from this area right here. As we're losing our myelin, you can appreciate the ghost white is gone. You get more of a smearing, an increase in the red and green, as the body is trying to upregulate the insertion of voltage-gated sodium channels in order to try to continue to propagate that signal. Otherwise, the signal will just stop at that location. So this is what's happening with multiple sclerosis. And I like this example because you won't be able to understand MS if you don't understand how it really works to begin with. That's why it's a great test question. Because I'm asking a practical question that many of you are going to come into contact with in your future careers or in your family or with friends. And if you can explain what's going on in a late-stage MS patient, then you have a good understanding of how an action potential travels. Does that make sense? Okay. Then after that, last week, you kind of finished up with where I started, which was the faster transduction happens down larger diameter axons, and slower transduction happens down smaller diameter axons. So if you have a larger diameter axon and it's myelinated, that would be the fastest speed. You with me? So on the exam, you would pick the number that corresponds with the fastest rate. Right? What does a rate mean? Come on, somebody answer the question. Measurement per time. So it has to be per some unit of time. Right? Like meters per second, kilometers per hour, miles per hour, those are all rates. Are you guys tracking with me? I'm really trying hard to give you guys hints for battling the weather, coming over here, listening to bad California jokes. Okay? I do apologize. I'm not trying to be down in California. It's just interesting how the water thing is coming out. All right. Any questions on that? All right. So now,
I have
question. Yes.
Why does the signal decrease when it's unmyelinated? Like, why does it diffuse?
So the question is, why does the signal dissipate when there's no myelin? Is that right? Is that accurate? Rephrase. So let's take this for example right here. If you lose the myelin and you don't put in a voltage-gated sodium channel, then this is not insulated. It's like having an uninsulated wire. And so that slightly negative charge and the depolarization that took place there, it's in millivolts, and it's very subtle. And it's only at the surface. And so what happens is it dissipates out of the cell, and you lose it. You lose the fidelity. Now the cell doesn't know, it's not enough to reach threshold in the next segment. Okay, so you have to preserve that electrical difference so that it reaches threshold in the next segment at the next voltage-gated sodium channel. And so if you have myelin, it insulates that signal and it pushes it downstream until it gets to the next voltage-gated sodium channel.
So how do some neurons work that are not myelinated, naturally not myelinated?
So the neurons that are naturally unmyelinated, like what you find in gray matter, is they have voltage-gated sodium channels throughout their entire length. All the way through. There's no gaps. So then the signal continues to propagate from one to another. It's like the telephone game. Do you guys ever play the telephone game?
Yes.
Anybody know what I'm talking about?
Yes.
I know you play this in California too, I'm sure. Right? So I whisper something into Maddie's ear, right? Like, I love California. Okay? And it comes out on the other side. Something totally garbled. Right? Or I like the rain and it's over here like, you are a pain or something like that, right? So the fidelity of that signal is determined by the ability to translate that information from one voltage-gated sodium channel to the next. So if there's a bunch of empty seats right here, right, that would be the analogy if myelin was missing in a myelinated axon. But the way that we fill that in is we have a long tube that she yells into the next person's ear, and then it propagates the signal. That's kind of what the myelin does. So in an unmyelinated axon, which is a great question, you have to have voltage-gated sodium channels throughout that entire row. Otherwise, you would lose the signal along the way. And that's why unmyelinated fibers are slower, because it takes time to turn those mechanistic pumps. And the faster myelinated fibers are quicker because there's these big gaps where you don't have anything mechanically moving. You just send the signal right down to the next person. Make sense? So that kind of a visible explanation of why one's fast and one's slow. When you think about a voltage-gated sodium channel, it is a pump in a way. It's a moving part. Something opens and stuff moves. It's passive because it moves down its concentration gradient, but it still takes time for that process to take place. So if you myelinate in between, you have a much quicker process. You're taking a shortcut. Does that answer your question?
Yeah, it does.
Okay. So now when we get to the end of the axon and we terminate, we actually have this structure that we looked at previously, which is the presynaptic membrane of the synapse. And we take this electrical signal and we're going to convert it into a chemical signal by releasing neurotransmitters. And the word, as it implies, is just that. It's a transmitter of the nerve signal. Neurotransmitter. And it's released into the cleft and then it binds to the other side to its respective receptor. And then it's going to trigger an action potential in some way, shape, or form to happen on the other side. And we'll talk about the ways that it does that. So we have an electrical synapse, meaning we have a gap in the electrical signal. And we have a chemical synapse, meaning that we use a chemical to get from one side to the other. Why do we set it up this way? Let me pose that question. Why waste time and, you just got done talking about speed. This seems slow. Why would we do that? Any ideas as to why you would design it this way? How is it advantageous? Is it slower than if this was just continuous? Yeah, it's a little bit slower. Now you got more moving parts. So what's the benefit? No thoughts? Yes?
If you have the brain's kind of like Legos, little bits in a row. If one of your little Legos goes missing or breaks or something happens to it, you can reroute around there.
Okay.
If it's one long continuous blob and a bit breaks, then the entire bit is broken.
Okay. Well, but even if you have lots of different pathways, I agree with you. You have these alternative routes, which is a good point. But all of those alternative routes have the same exact structure here. So you could still have those alternative routes, which is that plasticity that we talked about in the nervous system. So you're absolutely correct. But it still doesn't answer this sort of fundamental question of why bother making it this way, even in all those routes as well. Accuracy? Okay. Explain. Oh, okay. So if the electrical signal was just to transfer down, however, down the whole length of the axon would preserve the fidelity of the signal. It's always accurate all the way down the length of the axon. So these are just guesses. I mean, these are good guesses. Yes?
Control. It allows for specific control of which neurotransmitters are released and then taken up again. So that the nerve impulse doesn't continuously fire, causing a seizure or complex seizure, and therefore allowing it to fire with precision, if not just constant.
That was great. Did you guys catch that? Yeah, let's give a round of applause. That was really nicely done. So it's control, right? The body has the ability to control the response. So you can send the signal down the same way every time, even down alternative pathways. And it's accurate every time. But depending upon what neurotransmitter you release, you're going to get a different response. Okay? So now you have the ability to use the same neural network, just change the neurotransmitter at the other end, and it goes up in one situation and it goes down in the other. Okay? Nicely done. So let's look at some of these neurotransmitters. Let's talk about it. Well, there's a whole family of neurotransmitters. Talking about control, or another way of saying it might be even diversity of the signal. Meaning one signal can do many different things. And maybe that's a little bit of what you were getting at over there with rerouting. But we have over 100 known neurotransmitters. And these are chemicals that cross the synaptic cleft, that gap. That's what a neurotransmitter is. We're not going to cover every single one of these. We're going to cover specific examples within each of the families. The one that we're going to talk about the most is acetylcholine. And it's abbreviated ACH. Okay, so if you've been reading in the text, sounds like at least somebody has, right? You're going to come across acetylcholine, abbreviated ACH. We're going to talk about some other ones as the semester moves on, like GABA, which are in the family of amino acids, or monoamines, the family of catecholamines. We'll talk about two big ones. Actually, we'll talk about all three. A little bit of dopamine today, even. Epinephrine, norepinephrine, and dopamine. These are in the family of catecholamines under the broader title of monoamines. Also, a type of monoamine are serotonin and histamine. And then over here, as a neuropeptide, towards, I think later in the lecture series, we'll talk about enkephalins and substance P. These are neuropeptides that substance P is responsible for sending a pain signal. That's the neurotransmitter that's implicated when pain is detected. Substance P is being released. And then you've heard of these stories of like these, 103-pound moms that lift a car off of the baby stroller. You know what I mean? Yeah. Superhuman strength. They're not on crack or meth or anything. They're releasing a tremendous amount of what we call natural endorphins, and those are things called enkephalins, and they block the pain response. Okay, so you can see, based upon the great explanation in the back, having control, even just in one nerve, you have the ability to release a variety of different neurotransmitters. And that gives you the ability to make all sorts of different types of functional results. So how do the neurotransmitters work? They're either in two big categories. They're either what we call ionotropic or metabotropic. Now, ionotropic are neurotransmitters that directly change the membrane potential. So they bind, like acetylcholine is our classic ionotropic example. It's released, it binds to the acetylcholine receptor on the other side, and it opens the acetylcholine channel receptor has a channel down the middle, and that channel opens, and sodium rushes in. So when acetylcholine binds to the postsynaptic membrane, it alters the membrane potential of the other side directly. That's considered ionotropic. The way to remember that is, ions are going to immediately start moving. Ionotropic. Many of them, most of them, are in the family of metabotropic. Do I care that you remember which ones are which? I only really care that you know acetylcholine is ionotropic, and then many of the other ones that we're going to look at in the semester are what we call metabotropic. Metabotropic meaning like there's some metabolic process that happens first before it has its activity. Metabotropic. So a lot of times it's going to upregulate the metabolic production of cyclic A&P, which you know, you've heard, is a classic second messenger. Anytime there's a second messenger system that's involved with the neurotransmitter, it's metabotropic. It's going to have to do something within the cell before it actually triggers a change in membrane potential. But again, the end result is still the same. It's still going to alter membrane potential. It just does it using a second messenger system. Okay. The last little piece to finish up is this question for you. Saltatory conduction, action potential to jump from where to where? One astrocyte to another, an axon to a dendrite, one internode to another, one node of Ranvier to another, or one microglial cell to another? D. One node of Ranvier to another. Questions over this last little piece of Thursday's lecture?
How do they know what neurotransmitter to emit?
How do they know what neurotransmitter to emit? So the signal of the origin of the signal is going to stimulate that particular pathway to release that specific neurotransmitter. So for example, if it's a sensory receptor that's coming from the skin, and that sensory receptor is a nociceptor, that nociceptor is a pain receptor, and it's going to be programmed to release substance P. That's all it releases. Substance P binds to the neurotransmitter, and it sends its signal back to the brain, and it uses that same pathway to give the brain pain information. Okay? So it all depends on where the origin of the signal was. All right, we're going to move immediately into our third lecture. And I'm going to keep the camera kind of running so it all kind of runs together a little bit. We're going to start out with a couple of review questions. They're going to seem relatively easy because we just kind of covered some of these. After being released, neurotransmitters bind to receptors where? Postsynaptic membrane, neuromodulators, synaptic cleft, or synaptic vesicle. It releases from the presynaptic side and binds to where? The postsynaptic side. A, correct. A neurotransmitter attaches to its postsynaptic receptor, and this eventually induces the production of cyclic A&P. So we would classify this neurotransmitter then as ionotropic, metabotropic, inducing a change in charge across the membrane, two of the above or none of the above. What's the answer? So I think B is the best answer, but I think that this one potentially could be an argued test question if it showed up written the way it is. Right? Because metabotropic is what I'm going for, because it's cyclic A&P. But if you keep reading, eventually it induces a change in charge across the membrane, right? So how can I write that C question so that it is definitely not correct? How about directly inducing a change in charge across the membrane? You with me? So if C was modified to say directly inducing a change in charge across the membrane, then without a doubt the correct answer would be B. If you wanted to argue that it doesn't say that, so I picked two of these because it's B and C, yeah, I think you'd have a strong argument. Make sense? All right. So let's do a little review from Thursday. More review. I want you to draw action potential right now on your paper. And I want you to label all of the following. How's your action potential looking? This is going to be extremely helpful for the exam, by the way. What we're about to do, this activity. Okay, y'all done? Who's still working? Who's still drawing? All right, fine. We got time. Some of you just started because it was like, oh, before for the exam. Okay, I guess I'll draw it. You think there's going to be action potential questions on the exam? Okay. We ready? Did you label it? Yeah. Did you label it with the following? You know, you can just put a y-axis and put in the millivolts at the various specific locations. We're going to go over it. And with an arrow, tell me where depolarization is, repolarization, and what specifically is hyperpolarization. Ready? Okay. It should look something like this. So let's talk about that. So here's our y-axis. You can see that resting membrane potential, and you can write that right here, R&P, resting membrane potential, is sitting at about minus 70 millivolts. And so if you look upwards, you can see that the zero line is right here. And then it goes from negative to positive. So it goes minus 70 in shrinking numbers till you get to 0, and then in increasing numbers as you get past 0. Then you can see that threshold is labeled very appropriately at minus 55. And what are these little lines that look like they leave minus 70, but they don't, they depolarize, but they only get to maybe like minus 62.375 is what that looks like to me. I'm just joking, actually. But it's probably about minus 60, minus 62. You guys with me? And then it comes back down. And this one doesn't even look like it gets, you know, it's maybe minus 65 or something like that, minus 67. What are those called? What are they? They're failed initiations because they're labeled that, idiot, dummy. No, what's another name for them? A graded potential. Also known as what? A local potential. Because they're defined as an electrical deflection or a change in the electrical potential away from resting, but it doesn't reach threshold to stimulate what? If it reaches threshold, what is it?
An action potential.
It's an action potential. Are they failed initiations? Yeah, they're failed initiations, but they still are something, right? graded or local potentials. Now this one reaches threshold, and so now what is it called? You just said it? It's called an action potential. Which one of those, a graded potential or an action potential, is an all or nothing event? Action potential. What does that mean, Keller? All or nothing?
Once you're fired, it's fired and go back.
Once it hits threshold, it's committed to fire all the way through. It's not reversible. An action potential is not reversible. These are some of the types of questions that you're being asked on these worksheets. These are critically foundational, important things for you to know. The difference between an action potential and a local potential or a graded potential. So if you're wrestling with what we're talking about, and you haven't watched the lecture yet, go print out the worksheet that's in the lecture, by the way. Okay? And do that in preparation for SI. So this is considered the depolarizing side because we're making it less negative. Do you understand that? Depolarizing means it makes it less negative. If it makes it less negative, it moves it towards positive. It overshoots the positive, and then it comes back down, and this is repolarizing. It's making it polarized again. The word polarized, you and I are polarized on this issue. Does that mean that we think the same? No. Polarized means that you are on opposite ends of the spectrum. So if it's repolarizing, it's making it again more negative, away from positive. It makes it more negative, and it overshoots even the resting membrane potential. And then it slowly starts coming back to resting membrane potential. And we're going to talk about all the things along the way that cause this to occur again. Right? So let's do that right now. What is happening in the depolarization phase? What's taking place there?
Sodium is rushing into the cell.
Sodium enters the cell very quickly, rushes into the cell. It actually doesn't even take a lot of sodium to do that. It could be just a few molecules of sodium ion that come in, but they come in very quickly, right? Because they're positively charged, they're in high concentration outside the cell. Inside the cell is slightly negative, so the positive charge is attracted to the negative, and it's moving down its chemical gradient because it's high outside the cell and it's low inside the cell. So sodium comes in quickly because it's coming down its electrical and its chemical gradient. Make sense? All right, great. What happens right here? Some event must happen right here where it goes whoop, and turns around. Which gates close? Gates do close. What gates close? The sodium, the voltage-gated sodium gates close. Right up here, right? What's responsible for this? What gates are open, and then what's happening?
The slowing gates.
What's that?
The potassium slowing.
The voltage-gated potassium channels, which are much slower. So the slow voltage-gated potassium channels, they start opening kind of towards the top right here. They're open up here fully, and they stay open, and when do they close? Right where your pointer is. Yes. Okay? They start closing here, but they're slow to close, so they're not fully closed here, and that's why this overshoots. Why, if they're open, what is happening with ion flow in a repolarization event if you know that there's slow voltage potassium gates that are open? What's happening? Potassium is flowing which direction? Out of the cell. It's leaving the cell. Where is it typically high?
Inside the cell.
Inside the cell. Okay. And where is it typically low?
Outside.
Outside the cell. So is it leaving the cell in a similar electrical chemical gradient that sodium is?
Yeah.
It is? No. Right? Because potassium's high inside the cell, right? And it's leaving. So it's going down its chemical gradient, but what's the inside charge?
Negative.
Right? Well, it's slightly more positive, right? But eventually becomes a little bit more negative, right? So it doesn't really have an electrical gradient that it's really using. It's only using its chemical gradient. So it's going to leave the cell a little bit slower, and that's why this spike happens quickly and this one happens more slowly. Question?
Don't the potassium leak channels and the sodium-potassium pump affect the repolarization rate?
The potassium leak channels affect everything because they're always open. Okay? So it wouldn't be correct to say that they affect here because they're on over here too. They're just always leaking potassium. Okay? And the sodium-potassium pump is always on as well, but it always being on is what's responsible for bringing it back to a resting state. Because the sodium-potassium pump is going to sort of reorganize sodium location and potassium location. Okay? So it is correct to say that the potassium leak channel is always running, as is the sodium-potassium pump. But the potassium leak channel really doesn't do much to bring it back to resting state. It's more of the sodium-potassium pump that does that. Okay? So do you guys understand what we just walked through? If you're wrestling still with those topics, then you definitely need to go SI. You need to come see me in the office hours, and we need to go over them more. Now what I want you to do, we're going to start getting ready for the exam already, okay? I want you to put the following numbers on your graph. And this is something that you're going to, it's going to be remarkably familiar to you. So I think it's worth doing. Okay? Shouldn't take long. 1, 2, 3, 4, 5, 6, 7. Now what I want you to do is I want you to write down the number that best represents where this following event takes place. Almost all voltage-sensitive potassium gates are now closed. Almost all the voltage-sensitive potassium gates are closed. What number? Don't shout it out, but get ready. Who wants to volunteer a number? Explain why. In the back.
I'm going to have to say 4 because at that point the sodium gates become inactive for a second, allowing repolarization to occur so it doesn't propagate backwards.
Okay, you're going to say 4 because the sodium gates are closed?
That's when it's reached its the highest polarity.
Okay, but I'm asking about potassium gates. Okay, okay, so save that answer for a different question. Yes, over here. 6. Okay, why 6?
Because the gates are slow, so by the time it's hyperpolarizing,
Yep.
it's realizing that it needs to be shut.
Perfect. Okay, the slow voltage-sensitive potassium gates, not sodium in this question, potassium, are slower to close, so they don't close until 6 completely. Perfect. Okay, but I'm glad we're doing this because in the back you're totally right if we're talking sodium. But that's why we got to read these questions carefully, okay? So that's why we're doing this. Voltage-sensitive sodium gates are closed. All right, so this is for you back there. Voltage-sensitive sodium gates are closed, and they're inactive. And they're inactive. So what answer do you like? 4. You go 4. I totally agree with you. No arguments. Right? They're closed and they're inactive, just like he previously said. How about this one? What's the number where all the voltage-sensitive potassium gates are finally open?
4.
Okay, finally open.
4.
What do you guys like with finally open? 4? How many of you like 4? Do I have any 5ers? Some people like 5, but I think we would need to hone in the question a little bit more for a test question. But for a classroom exercise, I would say 4 and 5 are both good answers. The voltage-sensitive potassium gates are open in 4, and they are definitely open in 5, and they're closed in 6. So 6 would not be good. 3 would probably not be good because it's dominated by sodium voltage gates. So 4 and 5 would both be, I think, appropriate answers. How about this one? Write down the number where you have the 2 scenarios. Voltage-sensitive sodium gates are open, and voltage-sensitive potassium gates are nearly open. Nearly open.
3.
Ooh, Ooh, you like 3. You're over here with 3. Okay, very good. Subtleties to the question, right? But it really is begging the question, do you know the material well enough to be able to answer it? Okay, the point at which the process is still reversible. Where is it still reversible?
1.
Okay, 1. Why 1?
Because you're still in trouble.
Because it hasn't reached threshold, it's not an action potential, it's considered what? A local potential or a graded potential. Very good. How about this one? Cell is at resting membrane potential, but ions may not yet be in their proper location. Firing may not be possible. 6. You guys like 6? Okay. I like 6. I think 6 is 6. I think you could make an argument for the end of 6 or 7 is really where it's at resting membrane potential, right? And it's probably, if it's not possible to fire, it's at resting membrane potential, but it's considered what?
Inactive.
Refractory. You guys remember refractory period? Okay. So I would argue 7 is probably a better answer than 6, because 6, it's not quite at resting membrane potential. It's still a little bit overly negative. All right, here's a picture from a textbook. So did that make sense, what we just talked about, at least? If you didn't get them all right, are you at least following the questioning? And if you're not, this is where you need to really focus on how do I decipher these graphs and understand how the electrical signal corresponds to actual events. And those events mean that something physiologically is happening. So here is a different picture from a different textbook. This is from a Marriott text. And the reason I want to show you guys this is there are different ways to represent this, but we're all talking about the same thing. So here's our resting state, right? Here's our action potential. Here they label it 1, 2, 3, 4, 5, and then they go to 1. We had 7 labels on ours. Okay? It's not important that they're labeled differently. It's important that the events still make sense to you, even if you look at somebody else's explanation of how the process works. So let's just look at this. All right, resting state. We've got a sodium channel in the resting state, which is closed. All right, we have a potassium channel, which is closed. These are the voltage-gated channels. Here in the depolarization phase, which is 2, that's right here, as it's rising to threshold, you have one sodium voltage gate that's open, and the other one looks like it's closed. So sodium is starting to enter the cell. Once it hits threshold in 3, that's up here, both of those voltage-gated sodium channels are open, so sodium quickly is coming in. Over on the other side with 4, and they don't talk about what happens right here, which we did, but here at 4 you can see that the voltage-gated sodium channels are definitely closed, and the voltage-gated potassium channel is open, so potassium is leaving. That's repolarizing it down to 5. At 5, you get this undershoot, or this hyperpolarization, which is a much better term than undershoot. Okay, that's why we don't use the Marriott text, but that's okay. Again, the voltage-gated sodium channels are closed at step 5, and the voltage-gated potassium channel is still open. And they really should kind of show it maybe half-mast, if you will, like almost closed but not quite closed, because by the time it gets to 1, it's completely closed. Okay? So just a different artist's representation from a different author that I wanted you to see. So if we understand this concept of action potential now, and it sounds like at least the folks that are in this room, you have a pretty good grasp, at least on the type of questions I'm going to ask around the action potential. And so I think you should be feeling pretty good. If that wasn't too painful, great. If it was, still lots of time before the exam. But again, don't wait the weekend before. So our exam is on a Tuesday this time, right? And so that means the weekend before, if that's the first time you pick up this material, it is a lot of material to try to cover for a Tuesday exam if you haven't been doing anything along the way. Well, I want to talk with you a little bit about how we stop an action potential. And these neurotransmitters that bind, and I'm going to show you a couple of videos that help to illustrate some of these things. They only bind for about a second. And then they usually come off, or they're reuptaken, or they're enzymatically degraded into their constituent parts. Those are reuptaken and they're recycled. If no new neurotransmitter continues to be available, then the stimulus stops. So the control example in the back. One way to control the signal is also to say, okay, I want to actually stop before I run into the wall. And so you can turn it off by not releasing any more neurotransmitter. You with me? You also could block it at that location. You could say, you might release neurotransmitter, but I'm going to release a blocking agent so the neurotransmitter doesn't bind to anything. And of course, control also means, within medicine, that we have figured out, because we're actually pretty smart as a species, we figured out how to make drugs that actually bind to those receptors and can either stimulate them or turn them off. Okay, and cocaine is an example that I'm going to show you here in a second. So, I mean, not like really have cocaine on you, just a video. Okay, just since it's on video, I just want to make sure that's clear. Okay, how does this take place? Well, we can have the neurotransmitter come off the receptor and it diffuses away. That's how it can turn off. We can have the neurotransmitter be reuptaken, phagocytosed back into the presynaptic side. Or we can actually digest it, destroy it using enzymes into its constitutive parts. Like, for example, acetylcholine esterase is an enzyme that takes acetylcholine and it breaks it into a choline molecule and a esterase molecule. If we look at the examples that I have here, I'm going to show you a couple of these. These are different than what's on your slide because I had a PBS cocaine example and they took down the link. So I had to go search the internet for cocaine. Nobody showed up at my door. That was great. Like, why are you looking for cocaine here, Keller? All right, so let's take a look at this. Do you really want to? Yeah, I do. I wouldn't have pushed this button if I didn't. Okay, so let's talk about this. It's a quick video, but here is the one side, and here is the other side, and you're going to see that the neurotransmitter here is dopamine. Dopamine is a neurotransmitter that gives you a sense of pleasure. Okay, it makes you happy. Okay, it's released when things are going swell. Okay, it's a good mood neurotransmitter, and it gives you a sense of calmness and you know, things feel good, like that euphoric feeling of when things are going right and having a good day. Okay? If you're having a bad day, there's probably not a whole lot of dopamine being released. Okay? And in fact, in a lot of disorders, like with depression, we will prescribe a version of dopamine to try to elevate somebody's mood. Okay? That's how that works. Okay, so let's watch this video. I think there's audio too, but I'm not sure what they said.
Without cocaine, the reuptake pumps are able to reabsorb dopamine after it's finished having the treatment. But with cocaine, it shows here that the cocaine blocks the reuptake pump. Therefore, dopamine cannot be reabsorbed and it increases the feeling of pleasure due to the excessive amounts of dopamine.
Okay, did you catch that? Can you hear it? Uh-oh, this is going to be bad. Bottom left, where am I? Oh, replay. Quick. There it is. It sounds like...
Without cocaine, the reuptake pumps are able to reabsorb dopamine after it's finished having the treatment.
After dopamine binds, it's supposed to be reabsorbed. You add cocaine, which is the purple, okay, and it blocks the reuptake, and the dopamine sticks around and continues to activate that other side. Okay? So very simply, that's one example of a pharmaceutical that talks about how the neurotransmitter junction allows for this control feature. Okay, let's look at another one.
Our brains are composed of networks of billions of tiny nerve cells. Signals race around these networks, crossing the gaps between the nerve cells using chemical messengers. One network is dedicated to making us feel good when we do something that is essential to survival, such as eat, drink, or have sex. Normally, when this network is activated, the nerve cells in it release the feel-good chemical messenger, dopamine. Dopamine travels across the gap from one nerve cell to receptors on its neighbor, triggering a chain reaction which makes you feel good. This dopamine then returns to the nerve cell that released it for further use.
See how it goes back? Reuptaken?
Cocaine hijacks the feel-good network in the brain. It causes huge increases in the amount of dopamine throughout the network, flooding the brain with feel-good chemicals. And it also stops the dopamine returning to the nerve cells that have released it, meaning more and more builds up.
Cocaine blocks something called the dopamine transporter. And what that does is it mops up dopamine that sits in the space between two cells. And if that reuptake is blocked, what you get is an awful lot of dopamine floating around this space.
The flood of dopamine sends the feel-good network in the brain into overdrive, producing a sense of euphoria and self-assurance, making the user more animated and talkative.
Okay. I just showed you that you got a cool accent. All right. Questions over that? So there's another example that I want to show you that involves a video. And what I'm going to do with this one is I'm going to post the video on BB Learn, and I'll make an announcement when it's there so you can look at the activity of Botox. Okay? So I'm going to talk about it, and then I'm going to have you guys watch the video on your own. But did you have a question? Okay.
Yeah. What happens after that part where all the dopamine just builds up right there? Then what?
Like in the presence of a pharmaceutical, like
Yeah.
like with the cocaine example? So the cocaine has a half-life. So it'll stay in that synapse and cause that activity based upon the dose that the individual took. And eventually it's going to degrade. The cocaine signal will degrade. The DOPA reuptake transporters will become available again, and the dopamine will be reabsorbed. Okay, and then the individual will come down off of essentially the cocaine high. Okay, but there's lots of drugs. Cocaine is just fun because I get to say cocaine like 12 times in class. But there's lots of examples of pharmaceuticals that operate this way, right? And because we understand these pathways, we can build drugs, pharmaceuticals, to do certain things. How many of you had surgery? Whether you were asleep or whether you were awake and it was local. So general anesthesia would be you were asleep. Local anesthesia would be you were not asleep, but you couldn't feel a thing. Those are two different types of pharmaceuticals, and they behave very differently. Okay? The general anesthesia targets all over the body. Local anesthetics would be in a specific location. Okay? If you talk to your mothers, right? Some of them may have had medication when they were delivering you, and some might have opted not to have any. And so an epidural, right, is pharmaceuticals being delivered into that epidural space in the spinal cord, and it blocks everything distally or downstream. That's why they can still, punch your dad while they're pushing, but they can't feel their legs, okay, because it moves everything downstream. So Botox is another example. I use these crazy examples because they're kind of fun to talk about, but it usually helps you guys to remember them. Okay, a lot of students will remember, I don't know what we talked about, but he talked about cocaine for like 10 minutes. Okay? And then he moved on to like Wrinkles, okay, with Botox. So yeah, this is botulinum toxin that we're about to launch into. And this is a neurotoxin. It decreases the overall activity of muscles. So the wrinkles that we have on our face are in part due to basal level muscle contraction. Right? You ever heard that, your mom like, don't squint like that, don't like furrow, it'll stay that way. Well, there is some truth to that. You're strengthening these muscles, and then they're going to do that activity, and then those wrinkles will kind of permanently sit down in your skin. Now, there's other issues as we get older because elastin presence goes down, and you get wrinkles in other places, and the skin sags, and you have a heart attack, and you gain weight, and you get diabetes, and then you die. So don't get old. It sucks. But with Botox, the way that it works is we do these injections, and it's a nerve toxin. It prevents the release of acetylcholine at the neuromuscular junction. So that junction where the last example was dopamine, right? This is acetylcholine crossing. Acetylcholine is essential as a neurotransmitter to take the electrical signal to the muscle to say contract and wrinkle up your forehead. And so if you inject a neurotoxin there, you won't contract your forehead so it smooths out. Another example of this is, have you ever been around a stroke patient, a post-stroke patient? Okay? If they survive, and they're fortunate to survive, what are some of the features that you've noticed about them? Okay, they might have, if it was a right cerebral stroke, the left side of their face may be sort of slightly droopy, right? And not only is it slightly droopy, but it's actually wrinkle-free because they are no longer able to send motor neuron signals to the facial muscles on the left side, and that's why it droops a little bit, but it smooths out. Same kind of effect here. If you can't contract the muscle, you don't have that underlying muscle tone to create those potential wrinkles. Okay, so the video is quite interesting, but I've showed 2 videos already, and it's, I don't want it to be like, let's do the Star Wars trailer next, right? Which would be totally wonderful. If you haven't seen it yet, you should definitely crash the web again tonight. We should all do that. This is how it's working. All right? Oh, there it is. I was like, what did I do with my pointer? So here is a vesicle that's containing acetylcholine. Well, it uses this docking protein known as SNAP25. That's the name of the protein. The SNAP25 are these blue docking proteins that grab this vesicle and allow that vesicle to fuse with the membrane and spit out the contents into the cleft. The contents are acetylcholine. Well, what botulinum toxin, Botox, does is it actually breaks off this SNAP25 and it prevents these vesicles containing acetylcholine from ever binding with the membrane and ever fusing and dumping acetylcholine as a neurotransmitter into the cleft. So that is how Botox works. Now you can tell your aunt or your mom, okay? That's how Botox is working. Now let me ask this question. We all know that it's temporary. Why? Why is it temporary? What's that? Proteins can be replaced. This is a protein, SNAP25. This cell is still alive. So in a couple of months, the nucleus will have told the cell, manufacture new SNAP25, and that patient will have, again, the ability for acetylcholine to be released in the cleft. And so that's why the Botox injections only last 3 to 6 months. It's patient-specific, depending upon how quickly you manufacture new protein. Okay? Some people's nails grow faster than others. Right? Some people have protein manufacture that takes place more quickly. So that was our sort of group assignment, is why does it work and why is it temporary? Okay? But if you understand how Botox is working, then you're understanding how the cleft works, which is kind of cool. All right. In the last little time that we have today, we're going to go backwards a little bit and talk about graded potentials and local potentials. I want to talk about subsets of graded potentials, otherwise known as local potentials. Well, the subsets are that these local potentials, or graded potentials, could be a stimulatory signal And if it's a stimulatory signal, that means one can be sent, and then another excitatory or stimulatory signal can be sent quickly, and the two of them can summate. And if the two of them summate past threshold, what happens? Anytime I say past threshold, that should be like a clue. Action potential. So if you have an excitatory stimulus that doesn't cross threshold, but you get a second one that gets released and adds up or summates, and it crosses threshold, now it becomes an action potential. Now, in addition to that, if we had one that was inhibitory, instead of making the membrane less negative, what would it do? It would make it more negative. Okay? Those would be called inhibitory postsynaptic potentials. So we have excitatory postsynaptic potentials that are a type of local potential. These are types of local potential where sodium flows in, and when sodium flows in, it causes a slight negative deflection of that membrane. The neurotransmitters that most often are associated with excitatory postsynaptic potentials are glutamate and aspartate. The other flavor, if you will, of a local or a graded potential is what we call inhibitory postsynaptic potential, otherwise abbreviated as IPSP in comparison to EPSPs. Now the IPSPs, inhibitory postsynaptic potentials, they increase the negative charge on that membrane. And the neurotransmitters that are usually associated with this are glycine and GABA. And so you can kind of see how this works. Here is a axon. Here's a synapse right here, where you got the presynaptic side and we have the postsynaptic side. As it releases the neurotransmitter, you get an excitatory, there's the EPSP, excitation of the membrane on the other side. Now, add in this grayed out inhibitory neuron, and now it's live on the right. And this inhibitory neuron can actually release the neurotransmitter, usually glycine or GABA, and cause an inhibition of this neuron from firing onto the postsynaptic side. And this is how you would shut down the propagation of that signal. Does that make sense? So it's kind of like a brake control, if you will. Now, if we continue on this theme and we look at it in some slightly different ways, this is really 3 slides in a row showing really the same information or same type of information. So this is a diagram that is showing a postsynaptic cell. Here we've got an excitatory neuron in green, we have an excitatory neuron in black, and then here we have an inhibitory neuron in red. Okay? And if you can't see the colors, this one's labeled EX1, EX2 for excitatory 1, excitatory 2, and this one's labeled IN1 for inhibitory. Now, if you look at this tracing, this electrical tracing, we're going to introduce a concept called temporal summation and a concept called spatial summation. So temporal summation is just that. You sum the electrical signal over time. It's time dependent. Spatial summation means that the signal is coming from 2 different locations, spatially or in space. So if we look at this example up top of this postsynaptic cell that has multiple innervations, up until this point, I've only taught you that a neuron goes to another neuron. But in the body, it's not that simple. There's a neuron that goes to another neuron, and there's, as was mentioned over here, there are side branches. And there's other neurons that also come onto that same neuron, so that we can control that response. So if we were to fire, and that's what this is showing, here's minus 70, and we have a membrane potential on the postsynaptic side, and we fire the green one up top, excitatory 1, EX1, and then EX1 again, but they're too far apart from each other. Same neuron firing, too far apart, so it can't be spatially summated because it's the same neuron. It can only be temporally summated, but they didn't fire close enough together in time to reach threshold, so you got no action potential. If you put EX1 here and then you fire EX2 immediately at the peak of EX1, and you fire a second EX1 right at the peak of the first EX1, it reaches threshold, and now you actually get an action potential that was temporally summated. Because it was added together, summated, in time before the first one dissipated. Make sense? Okay.
Do these summate at the axon hillock?
Yes, they're typically going to summate at the axon hillock or that trigger zone is what we call it. Okay? Then this third example in C, here we got a lot going on. We've got an excitatory 1 firing and we have an excitatory 2 firing, but they didn't fire on top of each other, so there's no action potential. But just to the right here of this tracing, you get the EX1, the green one, and the EX2, the black one, firing almost at the same time. They're different neurons in space, so they added together to reach threshold and cause an action potential, so we call it spatial summation. Okay? Last example, introducing our inhibitory neuron. So here we can see that we fire our excitatory neuron in green up top, EX1, and then shortly after that we fire IN1, our red one down here that's inhibitory, and those signals don't do anything because they're too far apart. But over here just to the right, if you fire both at the same time, they cancel each other out. Okay? So temporal and spatial summation. Now the last picture that shows this information is maybe a little bit more familiar to you, because it's sort of a play on an old tracing where this is, I know, this is how well I draw with computer, put me on CAD, watch how bad it gets, right? So this, what are these little bumps called? Are they an action potential? What's that? They're graded potentials, and what type are they specifically?
Temporal.
Well, it's actually labeled conveniently in case you're curious. These are excitatory postsynaptic potentials, and they're drawn to try to demonstrate that they summate to reach threshold and to trigger an action potential. Okay? So, when we think about the circuitry of adding things together, we can organize the circuitry in different ways. The way that we organize the circuitry allows us to send different signals. So here's an example of temporal summation. Here's an example of spatial summation. Same exact concept that we just talked about, just shown in a different way. So you can see here's 3 separate neurons converging onto a single neuron. This one fires quickly. Right? That's what these lines are. Those lines are action potentials. Or, I'm sorry, are electrical signals. If they fire close enough together, then they summate at the trigger zone to fire an action potential. It's the same single neuron over time, so that's temporal summation. Here, a different example, you got all 3. You can see that the electrical signals are further apart, but they're coming from 3 different locations to spatially summate and trigger the action potential. Okay? Why is this important? Our last slide. We have different circuitry. We have different circuitry throughout the body. If you look at diverging circuits, we have an input that comes in to multiple outputs. One input coming in to multiple outputs. This is an example of diverging circuitry of what you would see going to a muscle. Right? Muscle contraction. Now in contrast, multiple inputs coming to one output. What do you think that would be? That would be sensory, right? Multiple inputs converging, sensory information coming in. You're getting lots of information to one output to the brain. Here's a different type of orientation of what we call reverberating, where you have an input leading to an output, but it stimulates this to go over and over and over again. And this reverberating is an example that we find in inhalation. When you inhale, you take a deep breath and you want to keep going and stimulate that muscle contraction again, that neural pathway to go over again and again and again. That's a reverberating circuit. Our last example, an input to a single output, but you have multiple ways to get to the output. This is called parallel after discharge. Where we see this, this is the example of, like, if you look at a light and then you close your eyes and you can still see that light, that's due to this parallel after discharge neuronal circuitry that we have in our retina. We'll talk more about that in our special senses lecture. Okay, I'll see you guys on Thursday.